There are two main methods for sequencing DNA. Chemical method, developed by Alan Maxam and Walter Gilbert. The chemicals are used to adhere the DNA at certain position, generating a set of fragments that differ by one near. The final fragment of DNA is relabeled with 32P at the 5 ends of each strand, and the strand denaturated, separated and purified to give a population of labeled strands for the sequencing reactions. Then a chemical modification of the bases in the DNA strand is done. It's done in series of 4 or 5 reactions with different specificities. Then the modified bases are removed from the sugar groups and the strands adhere using the chemical pyperidine. Large number of molecules and the different reactions will produce a set of fragments which terminate at different bases and differ in length by one nucleotide. This is known as nested fragments.
Enzymatic method, developed by Fred Sanger and Alan Coulson, involves enzymatic synthesis of DNA strands that terminate in a modified nucleotide. A copy of DNA is made by clean-out fragment of DNA polymerase. A single-stranded DNA and a primer is used to provide the 3 terminus for DNA polymerase begins synthesizing the copy. The production of nested fragments is by the incorporation of a modified DNTP in each reaction. DNTP lack hydroxyl group at the 3 position of the deoxyribose necessary for chain elongation. Those modified DNTPs are known as dideoxynucleoside triphosphates DDNTPs. The 4 DDNTP A, G, T, and C forms are included in series of 4 reactions. Each contains 4 normal DNTPs. Each R series produces a series of fragments terminating at the specific nucleotide and provide a set of nested fragments. The chain is labeled by including a radioactive DNTP in the reaction mixture. Analysis of fragments is similar for both involve Hell electrophoresis and autoradiography when radioactive label is used.
The microbe bridge reference database allows the CDC to use sequencing technology to quickly identify microbes and help stem outbreaks and epidemics. When the Ebola outbreak threatened West Africa, next generation sequencing technology enabled research to track the evolution of the Ebola virus in real time. Cancer up to 100 patients will have their tumors sequenced for genetic mutations that can be exploited by target therapeutics. Those patients will benefit from personalized genomic treatments options tailored to their tumors.
DNA sequencing plays a vital role in the field of agriculture. The mapping and sequencing of the whole genome of microorganisms has allowed the agriculturists to make them useful for the crops and food plants. For example, specific genes of bacteria have been used in some food plants to increase their resistance against insects and pests. As a result, the productivity and nutritional value of the plants may increase. DNA sequencing information is important to planning the procedures and method of gene manipulation. DNA sequences can be used to find a polypeptide sequence from the data bank or to compare with DNA sequences from other organisms for phylogenetic analysis.
The main application that had the chemical sequencing method was to know the interactions between the DNA and the proteins, and also to know the structure of the nucleic acids. Other of the main application of chemical sequencing is in the field of epigenetics, which is responsible for studying the mechanisms that regulate the expression of genes without have a genetic modification. Other of the applications for the chemical method was for being a fundament for the Next Generation Sequencing NGS.
Highly technological equipment to carry out electrophoresis of DNA and proteins, as well as the hybridization of nucleic acids and the quantification of the same through techniques such as spectrophotometry. The off-check DNA extraction kit is designed to perform the purification of human viral, bacterial, and genomic DNA from samples of human origin. The samples are lysed quickly and efficient by the addition of proteins. The lysis buffer and the ethanol create the appropriate conditions for the binding of the nucleic acids to the silica membrane of the columns. The carrier RNA improves the binding and recovery of viral, bacterial, and genomic DNA in low concentrations.
3500 genetic analyzer for requesting and fragment analysis. The 3500 genetic analyzer is an A-capillary sequencing instrument specifically designed to support the demanding performance needs of validate and process controlled environments while retaining the unsurpassed application versatility that life science researchers expect. The 3500 series data collection software supports sequencing and fragment analysis and integrates seamlessly with downstream software for secondary analysis of genetic data. Ion Gen Studio S5 Next Generation Sequencing System for targeted sequencing. The Ion Gen Studio S5 series is flexible designed to enable a broad range of target NGIS applications with industry-leading speed and scalability. Select from 5 different sequencing chips to sequence and throughput range from 2M to 130M reads per second.